during my PhD and postdoc. So the lab was sort of half neuroscience, half engineering, and this was innovative at the time and is still, pretty new, right? Which is, using engineering computer science principles and methods to further our abilities to do neuroscience, both in terms of understanding how the brain works, as well as designing devices to help people, which is the topic for today. Yeah, and then I started here eight years ago, and my group is trying to understand various parts of the brain, how they work, in terms of how many neurons interact with each other, right? So if you look historically at the field of neuroscience, people have studied one neuron at a time for many years. And there are 100 billion neurons in the human brain, right? So think like if you study one neuron at a time, how difficult that might be to understand how the brain works, right? And so about 15, 20 years ago, people started monitoring the activity of tens to a few hundred neurons at the same time. And so you can imagine that sort of opens the door to understanding things that were not possible to understand with just one neuron at a time. But just recording the activity is not understanding, right? That's just the first step. That's collecting the data, right? And then there's a lot more that's required in going from data collection to really creating new understanding about how things work. So that's sort of where my group, there are others in the field who work in this area as well, but this is where our focus is to really try to understand how the various aspects of the brain works, use in terms of how neurons interact with each other. So I think in terms of how it relates to this course, I think one of the exciting things in the coming years is really like neuroscience and sort of machine learning, deep learning has really evolved in parallel. And people have tried to bring the two closer together because it's well recognized that in principle, one can help the other, right? Right. So if the models that are used in machine learning and statistics could be inspired more by biology, that would be pretty cool. And then also to do neuroscience, we rely on a lot of machine learning and statistical tools, right? And so I think in the coming years, if there's more and more crosstalk, I think that would be great. It's just it's a very big divide between the two fields, right? And so I think a really important challenge is to try to bring them together in the coming years. Yeah, are we set to go? Okay, cool. Do you guys have any questions or want to talk about anything before we start? Okay, so I thought an interesting thing to talk about today would be brain-computer interfaces. So what is a brain-computer interface? Okay, so typically when you say you have a cup in front of you and you want to pick it up, then visual information comes through your eyes, is processed by the visual center of your brain, and then motor commands are sent down the spinal cord to move your arm. Okay? But due to injury or disease, that pathway might be cut off, right? And so we want to help these people by reading out activity directly from their brain, and then translating that into control signals, either for a prosthetic limb, a cursor moving on a computer screen, right, et cetera. Okay, so here is a cartoon video of how a system like this might work. So here is a cartoon person who is paralyzed. And what we can do is to record activity from their brain. Oops. Okay, so like this thing are the electrodes, right, which are recording activity from the brain. And then the activity comes out through this wire. and then you'll see it being passed through an amplifier and then into a computer. And then inside that computer, right, here are some machine learning algorithms, or here, that are processing the neural signals and trying to interpret them. And it converts them to movements of this cursor on a computer screen. Okay, so now this paralyzed person, rather than moving a mouse with his or her arm, because the person can't because they're paralyzed, but they can still think, right? And so if we can read out their neural activity, then if they think left, the cursor moves left. If they think right, the cursor moves right. Okay, this is the idea. Okay, so with a system like this, this patient would be able to regain some ability to interact with his or her, the world around him or her. Okay, so a system like this could be very helpful for a person. So here is a picture of one of the very first paralyzed people that tested one of these systems. So a system like this would be useful for a person like this with this sort of interface. So for example, if the person wants to turn on or off the lights, he or she would think left, right? If the person wanted to move the wheelchair, he or she would think right, right? And so one could think about designing various interfaces such that by thinking to move in different directions, that would allow them to interact with the world. Okay, so we'll start off by talking about how the different methods that are available to read signals from the brain. Okay, and then we'll talk about a couple different flavors of brain-computer interface systems. Okay, so there are many methods available for monitoring activity from the brain. So at one end of the spectrum are the so-called single electrode recordings. And these have been used for many decades and are the standard in the field. So this is an electrode that's implanted into the brain. And then the electrode tip sits outside of a neuron. Okay? And even though it's outside a neuron, it can listen in onto what this neuron is saying. Okay, so what this gives us is signals from individual neurons in the brain, okay, which is its advantage of this method. But the disadvantage is typically we can only record from one or a small number of neurons at a time with this method, which is limiting. Okay, at the other end of the spectrum, there are methods like EEG or fMRI, some of which you might have seen or even participated in before. So in a hospital, you might have been in one of these before. So with these technologies, you can see the whole brain, okay, which is great, okay, but you don't get single neuron resolution. Okay, and the reason why I'm showing you this spectrum is not to say that one method is superior than another method in all respects, but rather it's all about trade-offs, right, as in any engineering problem. Okay, so just as an analogy, let's say Heinz Field is the brain, and let's say that each fan in the stadium is a neuron. Okay, so then these single electrode recordings, okay, is like lowering a microphone into the crowd and listening onto one conversation. Go Steelers! Right? Okay. In contrast, right, at the other end of the spectrum, methods like EEG and fMRI are like having multiple microphones above the stadium where you can hear one side of the stadium cheering or the other side of the stadium cheering, but you don't get to listen in onto individual conversations. Okay. So what we would like to do, ideally, is to be able to monitor what many people in the stadium are saying, individual conversations, right? And to be able to do that, one way in which there are two types of methods that have been developed in the last 15, 20 years. On the left here, we have multi-electrode arrays. So rather than implanting one electrode at a time into the brain, we can have 100 electrodes at a time in the brain. Okay, on the right here, we have optical imaging. Okay, so the way this works is you can inject a sort of dye into each of these neurons, and that dye changes its fluorescence. Right? I could show, I don't have the video with me, but you could see it like a video, where each of these blobs is one neuron, and then it gets brighter or darker in a way that's proportional to its level of activity. Okay, so in both of these cases, we can monitor the activity of many neurons at the same time with single neuron resolution. Okay, so back to the analogy, this multi-electrode array is like lowering multiple microphones into the crowd to be able to listen in onto multiple individual conversations at the same time. Right? And then optical imaging is like having a really powerful camera above the stadium that is able to track the mouth movements of each person in the stadium, and then from that deducing what each person is saying. Okay, so using this multi-electrode array, we get recordings that look like this. Okay, so let me just tell you about this plotting format. Okay, so these recordings were done in a monkey while the monkey was performing some task. Okay, so I'll tell you about the task later, but all I'm indicating with this now is that the animal's doing something. Okay, so here on the horizontal axis, we have time. Okay, this is about 3 seconds in time. And each row here corresponds to one neuron. And each of these tick marks corresponds to an action potential or a spike. Okay, so the way in which neurons communicate with each other is through these action potentials or spikes. Okay, they're all or nothing, right? So the shape of the waveform, right, doesn't matter. Okay, what matters is the time at which they happen. Okay, and so that's why we can plot the activity of these neurons in this format. Basically, each of these tick marks just indicates for that neuron when did it give off an action potential or a spike. Okay, so in a typical experiment, we have 1,000 or 2,000 pages like this. Okay, because all of this corresponds to the monkey making just one reach. Okay, but in a typical day, the animal can make thousands of reaches, right? And so you can imagine, okay, here's the challenge, right? We want to look at this neural activity and try to guess where the monkey is planning to reach to, left, right, up, down, right? If I show you 1,000 pages like this, it would be kind of difficult for you to make out like what's important in this activity for determining where the monkey is going to reach to, right? Just because our eyes are not well trained to pick up on subtle features, right, in this type of data. Okay? But it turns out that there's really strong signal in here. Okay? So basically, if you make 10 rightwards reaches, right, so you have 10 pages of this, okay, versus making 10 leftwards reaches, right, the patterns of activity that you see across these neurons for rightwards versus leftwards reaches is night and day. Okay, so it's not, it looks like noise, but it's not all noise. Right? There's strong signal in here. Okay, and so this is what allows us to develop a brain-computer interface, because the goal of a brain-computer interface is to essentially read someone's mind, right? Basically look at the neural activity, and from that try to guess what movement that they want to perform. Okay, any questions so far? Yeah.
What if the neural activity is happening somewhere else in the brain where the electrode is not listening?
Right, so it's really important that the electrodes are recording in the relevant brain area, right? And so, as you're alluding to, it's true that many brain areas are involved. Like, so let's say just to see a cup and pick up a cup, there are many brain areas involved just for us to perform that simple action that we take for granted. And so what's important here is that we record from neurons that are involved in that process. And typically, the area that we record from is the motor cortex, in particular, the arm area of motor cortex, which is, can be identified from anatomical landmarks. So our brain has various folds, and those folds are highly stereotyped. So if you compared your brain and my brain, we would have the same folds. And so we can use those landmarks to identify where the motor cortex is and also where the arm area in motor cortex is. And that is where we record these neurons. And within the arm area of motor cortex, we don't really select, I want this neuron versus that neuron. We just go into that area and record from 100 neurons.
So this is something that's very much single use case. I want to be able to move my arm versus get someone to walk. It's more, we need the right arm area, the left arm area, the right leg area, the left leg area, the balance area of the brain, like
Right.
areas.
Right. So for each of those, we would want to go to a different part of the brain, right? Because for walking, we'd probably want to go to the leg area of motor cortex. For balance, there's yet another part of the brain, right? Yeah?
Is this method very sensitive to the placement of the electrodes, so like the exact nature of the signal? So do you have to like train the model differently for each person?
Yeah, right. So great. So there's going to be, we're going to be using statistical methods to try to estimate the subject's desired movements from the neural activity. And so we're going to need some training data, right, to estimate the parameters of our decoder, right? The decoder decodes the neural activity into desired movements. So even within the same subject, right, on each day, at the beginning of each day, we typically retrain our decoder because the signals that we get can change slightly from day to day.
Can we use like transfer learning to take what you learned the previous day and start that?
Yeah, yeah. So there's a lot of in common from day to day, but there are some small changes, and so we need to make sure that that is accounted for. Yeah. So yes. Yeah. Yeah. So we take all comers. So all of the neurons that we record go into our decoder. Yeah, so one could do some selection process, right? So for example, one could say, okay, look at each neuron and then see whether that neuron, its activity does a good job at distinguishing movements in different directions, right, as a pre-processing step. And then only include those neurons which do a good job at distinguishing between different reach directions. Sorry? Sorry, was that a question?
So it's like training the data on solely on a neuron's activity.
Yeah.
And see if that data control that.
Seeing if that data, what? What was the last part? I didn't hear.
See if that could do a good job.
Yeah, yeah. Yeah, so we typically take all comers, but we could sub-select the neurons at the beginning for just the best ones, and then using those. Yeah.
Why does the brain activity change from day to day, like within the day itself as well? Why is it?
Oh, yeah, yeah. There are changes within the day as well, right? Okay, so there are sort of two types of changes, right? One is that there are, the brain adapts over time. Like we learn, right? When we sleep, something changes in our brain, right? Okay, so that's sort of one type of change, right? The other type of change is that the electrodes can move relative to the brain, right? So you can think about, like, our brain is like Jell-O, right? And then you have these electrodes that are implanted. And so you can get these micro movements, like, because the brain is breathing. I mean, when we breathe, the brain, like, pulsates, right? Or the heart beats, right? Okay, so there are small movements there, right? And the electrodes can move relative. All it takes is a really, really small movement, and then now the electrode tip is next to a different neuron. Right? And so, yeah, we see changes over the course of a couple hours within a day, and we also see changes across days. Yeah, go ahead. Either.
Some kind of online learning approach be desirable for this kind of constant changing of data sources?
Yep. So we're working on that problem. So we have worked on that problem, and we're continuing to work on that problem to try to do even better. Yeah.
Just going to ask if neurons that are physically close to each other tend to have the same patterns, or?
Yeah, it depends on what part of the brain you're in. So, for example, in the visual cortex, there is well-established organization where nearby neurons tend to have more similar properties. In motor cortex, it's less well-established. Yeah. But there is this coarse organization that I told you about, right? That, there's an arm area, there's a leg area, right? There's organization there. But if you look at a finer level, like you're recording from a neuron, and let's say you look one neuron over, right? What is its properties? There's no, to our knowledge, it's not necessarily the case that that next neuron is really similar to that first neuron in motor cortex. Right? Yeah.
The brain is the 3D structure.
Yep.
The equipment to record is kind of
Great.
2D.
Yep. Great.
How you cope with this problem?
Yeah, great. Yeah. So there are different technologies of this flavor where we're recording from inside the brain. So there are these other recording technologies where you have a shank, like electrode that's a shank, that you have multiple contacts on that same shank.
Right.
It's just one big electrode, yeah,
Okay.
that you implant, yeah. So there, again, you can get tens to 100 neurons, usually tens, a smaller number. And the advantage there is that now you can have different depths. And the brain is organized not only in different areas, but also in different layers, right? And it's known, for example, like this layer receives inputs from a different area, whereas this other layer tends to send signals down the spinal cord to move your arm. So layer information matters, right? So oftentimes, when I go to a conference and I talk about my work, people say, do you know what layer you're recording in? Because then that matters for interpreting what we're seeing. So these electrodes, the length of these electrodes, yeah, you can ask them to manufacture them at different lengths. So the lengths that we use target the output layer of motor cortex, right, in hopes that those are the neurons that are most directly related to driving movement. But that doesn't need to be the case. We could also record from a different layer, right, and ask, how that influences, or how the properties of the neurons in a different layer are different, right? And so this is something that has not been studied enough, I think, in our field, that we need to do a lot more of, which is to understand, the interconnections between different layers and how information is processed, right, across different layers within one brain area.
But if you use longer needle,
Yeah.
it may damage some neurons when you put it into the brain.
Mm-hmm. Yeah. So typically, like with these arrays, like what happens is when you initially implant it, there are not great signals. But then if you wait a few days, then you start seeing really good signals. And no one knows for sure why that's the case. But the running hypothesis in the field is that the brain views these electrodes as like a foreign object, right? And so there's probably a bit of tissue damage at the beginning, but then over the course of a couple days, then the brain sort of repairs itself, and then it starts, it comes back, right? But if you look at, like, in terms of like the function of the brain, so it's pretty incredible how much of the brain, like this is like not studies we've done, but in hospitals, right? When people have tumors, for example, right? It's incredible how much of the brain you can remove, and the person still seems like exactly the same person, right? Like you can, yeah. I mean, it depends, right? So if you remove like the motor cortex or the visual cortex, then those are really essential areas you might not be able to see or move anymore. But yes, highly, right? So then you can imagine, okay, with these electrodes, you implant them. They're going to affect some of the neurons along the way, but that's a pretty small number. Like there's no measurable effect on the subject's ability to do anything in the world from the first moment these electrodes are implanted. Like, yeah, there's no There's no measurable effect. Mhm. Yeah, it's kind of interesting, right? Because if you think about like the engineered systems that we design, like take a computer, right? If you remove a transistor, right, that kind of will probably mess things up, right? Whereas like the brain, you can start removing a neuron like however many you want. You're not gonna, like people are doing experiments now, not in humans, but in like small animals, right? Where you can like zap the neuron to like during the experiment, you have like, you know, a real-time view of like the arrangement of the neurons, right? And there are ways you can zap the neurons to like kill them, right? And you can zap lots of neurons, and behaviorally, you know, the animal is, you know, like in a fish, right? Behaviorally, the fish keeps swimming, right? Right? And so you connect that with, you know, these surgical findings that I mentioned, where people have tumors, right? And you got to have different parts of the brain removed, right? And it's amazing, like how much redundancy there is in the brain, right? Which is very different from the systems that we design and engineer. Cool. Okay, so we work with monkeys, and the reason why we work with monkeys is for these brain-computer interfaces, right? We want to have a direct translation route, right? So monkeys have brain structures that are very similar to ours, and they also make eye and arm movements that are very similar to ours. And so if we can develop a brain-computer interface that works well, right, with monkeys, then we have a direct translation route to humans. Okay, so there are two primary flavors of brain-computer interfaces. Okay, one is a discrete variety, and one is a continuous type. Okay, so the discrete type is like typing on a keyboard. Okay, so what matters? It's like you have a bunch of keys, and you're gonna they're discrete choices. Okay, the continuous type is like if you want to move a robotic limb or you want to move a computer cursor, is that you need to move continuously in space. Okay, so let's talk about the discrete case first. Okay, so here's a picture of a person who is paralyzed from the neck down, and she was equipped with one of these systems, and she was able to type out these messages by using this keyboard, right? And so this is ongoing work in the last few years at Stanford, where they've been getting really good performance with this person being able to type out messages. Okay, so I'm going to be telling you about sort of the basics about how one of these systems works, right? So it's based on the concept of motor planning. Okay, so what is motor planning? So when you're out on a picnic, let's say a fly appears on your picnic table. Okay, so typically you don't swat at it right away. You typically pause for a moment, figure out where the fly is relative to your arm, and then you swat, right? So what is your brain doing between the moment when the fly appears to when you start moving your arm? Right? There are a bunch of computations that go on in your brain. And it turns out that the activity in your brain during that split second is highly indicative of where your arm is about to move, okay, even though your arm hasn't moved yet. Okay? We call that motor planning. Okay, so in a laboratory setting, we don't have people swatting flies, but instead we have monkeys touching targets on a computer screen. Okay, so the way this works is that the monkey starts with his hand at the center of the screen. Okay, then we present a peripheral target here. So this is like the fly. So the monkey is trained to know that when the fly appears, he's not allowed to swat for it immediately. Okay, but rather he needs to, but he's allowed to plan to swat the fly. Okay, it's only when he's given a go cue, which is signaled by the disappearance of the center target, is he allowed to. Okay, so there's the go cue, right? The center target disappears. Now he's allowed to swat the fly, right? And if he does that successfully, he gets a juice reward, and then the whole thing starts again. Okay, so let me show you a video of this. So here's the monkey, his hand's like here, right? His fingertip. He's touching the center target, and the fly just appeared. Okay, so I'm going to show you this video. Okay, all the videos that I'm showing you today are in real time, right? And each tone is a juice delivery. All right. Any questions about this? Okay. All right, so while the monkey was performing this task, okay, we recorded from the premotor and motor cortices. Okay, so now you know what this top illustration means, right? So we have a timeline of this task. Okay, and the hand doesn't move until after the go cue is given. Okay, then we have a hand movement. Right? So most of the neural activity that we're recording is during a time when the hand is not moving. Okay, and we record neural activity that looks like this. So what we're going to focus on is neural activity during this so-called planning period between when the fly appears to when the go cue is given. Okay, so what we're going to do is to count the number of spikes that each neuron produces within this window, right? So like we're going to have a vector, right, where neuron 1 produced 10 spikes, neuron 2 produced 5 spikes, neuron 3 produced 12 spikes, et cetera, right? And that is the feature vector, right, or the data, right, that we're going to use to do a classification. Okay. All right. So just to show you in sort of cartoon form or picture form how we do this classification. So here is a hypothetical neuron where, okay, so what you're seeing here is horizontal axis of the animal's workspace and vertical axis of the animal's workspace, right? So this is like, this square is like the animal's workspace. Okay. The color map corresponds to the level of activity that this one neuron would produce if the animal were planning to reach to that location. Okay, meaning if the animal were planning to reach to this rightwards target, that this neuron would produce around 70 spikes per second, purple. Whereas if the animal were planning to reach to this leftwards target, this neuron would produce around 40 spikes per second, green. Okay, so then what we can do is to map each of these targets to a letter on a keyboard, right? And now you can imagine typing out messages by thinking of different targets. Okay, does anyone see a problem here, though? Sorry? Okay, we can do that. That's not a problem. Okay, let's say that this neuron produces activity of 40 spikes per second, which is green. What letter does it correspond to? It could be, conceivably, be G, A, B, C, or D, right? So how do we distinguish among those possibilities? Great, yeah. So this is just one neuron, right? Unfortunately, we do not have this scenario where all of the neurons have the same response properties, right? Instead, we typically have this scenario, right? So now what we can do is to look at the responses for each of the neurons and then compare them to the known properties of these neurons, which is like this color map, which we would characterize ahead of time. And that would allow us to triangulate, right, what the intended reach direction, or reach target is for the animal. Right? This is showing in pictures what a classifier does, right? So you guys are probably well familiar with classifiers given your background. So you can choose your favorite classifier, and it would likely work with these data because the signal is so strong in terms of which of the 8 targets that the animal is planning to reach to. Okay, so typically, what we use is a Poisson naive Bayes classifier. Okay, so Poisson because our observed data are counts, right, number of spikes, right? And so typically a Poisson distribution is a good first order descriptor of the spike counts that we observe. Right? So each neuron would have a mean of that Poisson, right, where that mean value depends on which neuron it is and also which of the 8 targets it is. Right? So the number of parameters in this case would be number of neurons by 8 in this classifier. So that's the first place we usually start, and then you can get fancier, right, to do a little better. Okay. So to sort of now think about the bigger picture of how this fits into what we want to do. So let's say that the animal is planning to reach to this upper left target, right? And then the activity that we record is a pattern like this across the array, right, where the color corresponds to the level of activity of each of the neurons, right? We then compare this pattern of activity that we record to the proper firing properties of these neurons. This box is the classifier, right? And the output is estimated target, right? And in this case, it is correct, right? Because the output of the classifier matches the target that the animal was planning to reach to. Okay, so now, as part of the experiment, the monkey is planning to reach this upper left target. We record the neural activity. We get this pattern of activity. We put it into the classifier. Out comes an estimated target. Okay, and then we present that estimated target back to the monkey, okay, by placing a circle at the location that was estimated by the classifier. Okay, so if all goes well, the circle should land on the fly, right? And if that happens, the animal gets juice. Okay, but it's possible for the estimated target to be here, say, right, which would be incorrect. Okay, and the animal does not get juice, and then the next trial begins. Okay, any questions about this? Yeah. It's water usually, but sometimes they like something with sugar in it. So I don't know, grape juice or or whatever they like. Yeah.
What if the estimate is always incorrect, but the monkey learns to compensate in a way so that the two land on the top of each other?
Great. Yeah. So this is something that we're like now. So this is a study we did years ago, but the studies we're doing now are focused on that question, which is how does to use brain-computer interfaces to help us understand how the brain learns. Because just like if I give you a tennis racket and I let you practice with it over the course of a couple weeks, you're going to get better at playing tennis. Similarly, when we give a subject a brain-computer interface and allow them to practice with it, they're going to get better at using it over time. So then we can look at what changes in the brain while they get better at using it, and that tells us things about how the brain learns. That's what we're doing now. Yeah. Was there another question right here? Yeah.
If you transform this movement to the speaking area, let's say if you want to say a word, people have to prepare before they speak a word. So could you recommend that neural activity before people speak? Is there an experiment on?
Yeah, people are working on that. Yeah, that is a more difficult problem. Just because the possibilities, right? If you think about different words that we can say, so I don't know what the state of the art is there in terms of what is the best someone can do, but people are trying. Okay, so let me show you a video of this in action. Okay, so what you're going to see, is a video that's similar to what you just saw. The key difference being that on many of the trials, the animal doesn't need to move its arm, and it can get juice. And the reason it can get juice is because of what we just talked about, right? Because we're looking at the neural activity, and we're classifying the neural activity. If the classification is correct, he'll get juice, and he doesn't need to move his arm, right? Okay, so in principle, we could just have him do this task without ever moving his arm anymore, right? He could just, one trial, like every trial is a so-called brain-computer interface trial. Okay, but then we tried that, and we ran into a problem because he kept getting these trials correct. And then the monkey thought, oh, I'm getting free juice. I don't need to plan to make arm movements anymore. So if the animal doesn't plan to make arm movements, the system doesn't work, right? This whole thing relies on him planning to make a movement, and then we can classify that neural activity. So that is why every now and then we need to give him the go cue to move his arm. So on every trial, he needs to believe that there's a chance he might have to actually move his arm, right? And that's how he can plan. That's how we can get him to plan, right, to move. Okay, so I'm gonna play this video. You have to look carefully, right? So here, right, is the fly, right? And so you're going to see that there's going to be a circle that appears around it, and that would be a correct trial, right? Okay, so based on the performance here in terms of how quickly we go and also how accurately we can classify, we can compute a bit rate, right? So like, let's say we have 8 targets, and let's say every selection is correct, right? Then that gives us 3 bits per selection, right? And then depending on how many selections we make per minute, that gives us bits per second, right? Okay, so the performance that you just saw here is roughly 2.5 bits per second, which translates into about 5 words per minute typing. Okay, but I'm showing you the slow version of this video so that you can see what's going on. Okay, now I'm going to show you a faster version of this video. Okay, so this corresponds to 5 bits per second, or typing around 10 words per minute. So now you're going to have to look really carefully because it's going to go faster. Okay, so what's going on here is the animal is able to make about 3 choices per second, right? So he's like, plan right, plan left, plan up, right? All within 1 second. And then we can classify each of those things in real time, right? So that's sort of the speed at which we're seeing here. The fastest that we were able to get in all of our experiments was 6.5 bits per second, which is typing around 15 words per minute. So it's a little faster even than this video. So it's not a modem, right, even, but it's something, right? It's, for someone who's paralyzed from the neck down, something like this could be very helpful. Okay. Yeah, did you have?
On the video it said 2004, right? So presumably in like 10-ish years, has there been significant progress in this?
Yeah. So I mentioned at the beginning there are different things we might want to control, right? So one is typing on a keyboard. So in terms of typing on a keyboard, this is still state of the art. Yeah, and the reason is because the reason why we weren't able to go faster than 6.5 bits per second is not because our system wasn't good, but it was because we were running into fundamental limits on how quickly the brain can think left, think right, think up, think down, right? So we were already doing as well as one could do, I think, with this. Where a lot of innovation has come in the last 10, 15 years has been doing things that are a little more complicated. Okay, so I'm going to show you videos of moving a cursor continuously on a computer screen. Okay, so that has been a harder problem, and we've made a lot of progress there to the point where now, just in the last few years, we've come to a point where you can control a cursor with your brain almost as well as you can control a cursor by moving a mouse with your arm. Right? So I'm going to show you a video of this brain-controlled cursor, which, to my eyes, looks very similar to what someone would do with their arm, which would not have been possible, in the mid-2000s. Another thing that people are working on, which I'll also talk about, is controlling robotic limbs. And robotic limbs have more degrees of freedom than a two-dimensional cursor moving on a computer screen, and that is still a hard problem today, right? To be able to make well-coordinated grasping movements and manipulate objects, that is still a major challenge for these brain-controlled systems. Yeah?
The reason that we can type much more than 50 words per minute when we actually have farms is because we have kind of motor memory, right? So we just plan like on a higher level that we want this word, and we make them work. Is that how it works? Yes.
Yeah, you're saying what about the mismatch between?
So like 15 words per minute isn't very fast when like we can actually type with our hands.
Right. What is the fastest that one could type with our hands? How many words per minute typically?
Like
100,
100.
right? Okay.
So
Right.
It's also, you're not just moving an arm, you're moving 10 digits while fate.
okay.
Right.
So it's like, I guess you've got 8 electrodes, right? And each one was planning its next move. Would that
Yeah, that would be really cool to think about, right? So, yeah, how do we get these systems to type at 100 words per minute, right? So we're only recording from 100-ish neurons, right? What if so let's say we could record from all the neurons in the brain. Does that help us get up to typing 100 words per minute? I want to say it has to be possible, right? Because our brain can do it, right? I mean, it has to be possible at some level. So I think, I think one thing you're pointing out is still there's a lot of room for good work to be done here. We're not even close.
Because the monkey there could use 2 hands. It's only 1 hand game. Right? It can use 2 hands. Maybe it's good.
Yeah, one could even say it's using 1 finger, right? I mean,
This is analogous to like typing one
Yeah,
key at a
maybe.
time on the keyboard.
maybe, yeah, right.
I
Mm-hmm.
think you are trying to make monkey type with finger. Why don't you make monkey just like sync A, sync B, type like that?
Like thinking about
Oh.
language.
The actual letters.
A monkey thinking letters? No.
Oh, you can use the Morse code.
I think it's hard.
Yeah, so we can design like a code, right? In fact, to estimate this bit rate, like we use like this equation with channel capacity, right? So given this particular like pattern of errors, like we look at the confusion matrix, and then there's an expression from information theory that allows us to calculate the information capacity, right, of a channel that has that sort of error pattern, right? So that assumes that we can come up with the best code, right, to do this, right? So that's already, in some sense, taken into account in this number. But yeah, I mean, you're absolutely right. Yeah, we can come up with some code, right, that corresponds, different directions, like left, left, right corresponds to one letter, right? And then right, up, left corresponds to another letter, right? We can definitely do that, right? But at the end of the day, what this still relies on is our ability to accurately classify different movements quickly, right? Because once we can do that, we can design any code around it. So I'm not sure I answered your question, though. So monkeys can't think about letters, but what we're, yeah.
Other ways like images of pictures, letters.
Right. Right.
Just like those thinking about the Chinese words, more like images instead of letters.
Right. Right. So, yeah. So I think that's a great idea in the sense that if we could train animals, or maybe one day we could do it with people, right, using some other recording method, right? The tricky bit there is now we should not record in the motor part of the brain, but we need to be in the part of the brain involved in thinking thinking about different visual forms, right? And it's not because, OK, so we can't be, OK, so let's say, can we record in visual cortex to do this? The answer is probably not, right? Because visual cortex is showing activity related to what we're seeing in this world. But if you want to type out a message, you need to be able to you need to be able to change that activity in that area volitionally, right? And visual cortex is usually locked to whatever is going on in the outside world, right? So we need to be in the brain area which changes the activity according to thinking about different visual forms. So it's not, first of all, it's not obvious which brain area that is. And then second of all, if we knew what brain area that was, I think the decoding problem would be pretty challenging, but it would be fun to look at. Yeah, so the reason why a lot of people sort of work in the motor cortex is because it's very concrete. It's not abstract, right? You're thinking about moving right versus moving left, and things are a little clearer. But I think as time proceeds, we should proceed into those more abstract brain areas and abstract classifications. OK, what time is it, by the way? Oh, OK, great. All right, so now let's transition onto the second part, which is going to be a little shorter than the first part, which is now doing this continuous control, right? So if you want to control a computer cursor on a screen or a robotic limb, right? It's not discrete choices anymore, but it's continuous control. OK, so here is a video of a monkey controlling this robotic arm using its neural activity to feed itself marshmallows. OK, so this work was done in Andy Schwartz's lab right here in Pittsburgh. Right, so in this setup, that same electrode array is recording from the motor cortex in this monkey, and that's what's controlling this robotic limb. OK, you get the idea. OK, and then here is a more recent video of a person who is paralyzed from the neck down, controlling a robotic limb, just using her brain. Okay.
We came back to see whether she would be able to control the robotic arm with nothing but her thoughts. They plugged her brain into the computer, and this is what we saw.
I can do it up and straight down and left and right and diagonally. I can close it and open it, and I can go forward and back.
That is just the most astounding thing I've ever seen. Can we shake hands?
Sure.
No, really.
Yeah.
Like, come right over here?
Yes, you can right
Okay.
there.
Oh.
Press your hand there. There we go.
Oh my goodness. Wow.
And I can do fist bump if you'd like.
That's amazing. What are you doing?
So this video represents state of the art, right, in what we can do with a robotic limb today. So generally speaking, the arm can move in the right directions, right? You can get the hand to sort of all open or all close, okay? But that's about it, right? And so challenges are like picking up objects of different shapes. Like that's still pretty difficult. I mean, this lab here at Pitt actually is working on this, and they've shown some results. But in terms of comparing to what we can do with our natural hands, like what we have now with these brain-controlled robotic limbs is not there yet. Okay, and then here is a video of state-of-the-art cursor control. Remember how I mentioned, there we have cursor control that's almost as good as moving a mouse with a hand. Okay, so here's the video. Okay, so here's a monkey controlling that gray circle just by thinking to hit the other targets. Right, so if you measure the speed and accuracy of these movements, it's 50 to 80% as good as what we can do with our arms.
And it's monkey.
Monkey, yeah.
And monkey will get juice when.
Yep. After each one of those, right? After it touches each of these targets.
But I mean, when you get juice and when you activate it, it has some latency.
When you activate what?
For example, you give monkey juice,
Yeah.
and he feel happy.
Yeah.
Between these two events, it has some latency. Right?
Yes.
So that latency affects the throughput.
Right. Yeah. So so we want to try to keep that time as short as possible so that we can start the next trial, right?
Yeah.
So here he's getting juice after each reward, but you almost can't even see it because it's happening so fast. It's just a split second. Yeah. So when we design these systems, we want to keep that time short so that we can go fast, right?
Yes, I mean the limitation must have. It has faster speed. It can apply to human.
So you're saying because with humans we don't need to give them juice between each, then we can go even faster?
Yes.
Probably, yeah. Yeah.
Is there any notion of whether, so presumably at the time when the monkey's thinking about this, because it has juice, it's like happier, right? Is there any notion of that maybe changing the signal that you would see? Like perhaps instead, imagine the monkey would still be following or performing this even if it was sad, right?
Mm-hmm.
Is there any notion of whether that would change what you're?
Yeah, I mean, we have a notion of being motivated versus unmotivated, right? So there are some days where the monkey, is unmotivated, right? Yeah.
But do you see changes in, for example, the readings that you get out of your electrode or whatever?
Right, it's a little hard to tell, right? Because if he's unmotivated, then we can't really know whether, the reason why, we can't classify very well is because he's not even trying to plan to make these reaches? Or is it because he is trying to plan the reaches the same as before, but the signals don't look as good? So my guess is that that for the most part, the signals in the motor cortex are going to be largely unaffected, but not completely unaffected by motivation. But there are other parts of the brain which are more directly related to motivation, and those areas will see very different activity for different levels of motivation. Okay, and so in terms of where we've sort of already talked about some of these things, right? Where is the field going, right? So I think the things that we've talked about today indicate proof of concept that these systems can work, and we are made some pretty good progress in the last 20 years in trying to get these systems to a point where they would help people substantially. But there still remains a lot of work to be done, right? And we've highlighted some of those. And so other ones that we haven't highlighted are things like, so the typical systems that we have today require a wire, right? You saw in the cartoon, there's a wire that carries a signal outside the brain. And so if you want a person to be able to roam around the world and use one of these systems, it needs to be wireless, right? And low power that they can carry on their head, that doesn't get too hot, right? And because of all the data that's streaming out, this is something that needs to be improved on, right? So people are developing wireless systems for recording and transmission that's tailored for this type of signal, right? because you want to make it as efficient as possible, both in terms of power and also transmission speed. Right? And then another thing that people are working on is the duration with which you can record with these devices. Right? So when you implant these electrode arrays, the brain thinks that it's a foreign object and starts building up scar tissue around it. Right? And so over the course of many months, and maybe if you're lucky, 1 or 2 years, then at some point you no longer can see signals on these electrodes because it's lots of scar tissue built up around it. Right? And so if you want these people to be using these systems, they've got to last 5 years, 10 years. Right? And so people are developing better materials, more biocompatible materials, so that the brain, when these electrodes are implanted, the brain views it as part of its own, right, rather than trying to reject it. Okay, and then, while all this sort of work towards moving these systems that we develop in a research laboratory with monkeys to human patients is extremely important and is something that we're interested in. The focus of my group here is not that, but rather using brain-computer interfaces as a tool to understand how the brain works. Right? So it turns out that there are many advantages of using a brain-computer interface to study the brain that we're able to address questions that are currently difficult to address using other methods. Okay, so that's it, and I'm happy to continue to converse with you guys, answer any questions. Yeah?
It seems like the neural network got many benefits in the early days from the neuroscience.
Yeah.
Nowadays, it seems like the deep learning are more engineer-oriented.
Mm-hmm.
The neuroscience, the tool field are far
Yep.
before. So how do you comment this phenomenon?
Yeah. I mean, so I don't know if it's farther than before. I would say it's always been far.
Okay.
Right?
So okay.
And I think, because I have sort of a foot in each, I think it would be nice to bring the two closer together. I feel like we have a lot to gain from each other. But at the same time, I understand why the gap exists. Right? I struggle with this, maybe struggle is too strong of a word. I think about this, on a regular basis, right, for our research, right? Because, we're doing work that can be, published in a neuroscience venue. We have work that can be published at NIPS or some machine learning venue, right? And usually we have a target audience in mind, right? And it's usually, either the neuroscience crowd or it's the machine learning crowd, right? But not like both, right? Because it's very hard to write a paper, right, that is going to be of interest and value by both communities, just because those two communities are so far apart, right? And so, yeah, I think this is a challenge and also an opportunity, moving forward. And I think one of the reasons why the two have been so separated is that we just don't know enough about the brain yet to be able to, we don't have any principles. Like, if you think about the history of physics, right, versus the history of neuroscience, right, where we are is pre-Newton, Copernicus, right? We're way early days right now, right? We don't even have there's no principle that I can tell you, the brain, that we have firm footing. We know that there is this very basic principle that the brain uses, and then we're going to use that to try to understand more what the brain is doing, right? There's not a single principle. People have hypothesized lots of stuff. People have tried to test stuff. Things have been controversial, but there's not a single principle that most people agree on that the brain works by, right? And so without that, I don't know how we can tell someone who's designing an artificial system that you should incorporate this principle into your system. Right? And so I feel like part of it is for neuroscientists to, we need to make more progress first to understand the brain before we can start building them into these artificial systems. But at the same time, I don't think we should just wait around, right, and say, okay, well, you neuroscientists, you should figure out more about the brain, and then we'll start talking in 100 years, right? I'm not sure that that's the best strategy either, right? And I feel, so there are a good number of people in machine learning who know a lot about the brain and are interested in the brain and go to, neuroscience conferences or can converse very well with neuroscientists. And I think there's a lot of value in those people as being, the bridges, right? And, trying to, like, even if there is not a principle that we all agree on, maybe there are some, interesting findings that, from the brain, even if they're controversial, right? That we can start building into these artificial systems and seeing if they help these artificial systems, classify better or, whatever else, right? And if they do, great. I mean, it doesn't have to mimic the brain, just needs to do a better job at, doing the task that it needs to do, right? And if we can use things that people are finding in the brain as inspiration for new ideas of, how to design new artificial systems, great, right? So I feel like, yeah, this is like an important area that we should all, try to strive towards. I think there's value. Yeah.
Suppose that there were like two electrodes placed in very different parts of the brain, and superficially they record very different signals, but on like a very high level there's similarities between the two. Like you're recording speech data and also writing data, and they both have like a root in language. Are there kind of models that use both of these different signals to improve this kind of?
Both? What are the two?
Like two signals that are superficially different, but they have like a common root. Like both writing and speech have like information about language within them.
Mm-hmm.
So are there approaches that kind of use both information sources to improve a common model and then branch off from that to address the two different tasks? Or typically these two sources are just treated independently for two different tasks?
Right. I'm not super familiar with the language stuff. Yeah, so I see what you're saying. And the areas involved in producing language and interpreting language have strong connections, the brain areas involved, right? So, are you saying, like, can we record from both of those areas to
like basically use both of those areas to improve this kind of shared model?
Yeah, I would hope so. Okay. Yeah, I'm not entirely, like, I'm not sure if anyone. So using this type of recording technology is a little tricky to get a large number of electrodes into the language centers of the brain because they're very deep in the brain. So most people who have done this sort of study has been with fMRI. And so there, with fMRI, you can see, the whole structure, but then you don't get single neuron resolution, and also the time resolution is very slow, right? So, and I'm sure people have, tried to study the interaction between the areas, and tried to leverage both. I don't know what the state of the art is.
The kind of concept applies to motor. Like there could be higher level concepts of motor movement, like running via crawling, and using different recordings for the hands or the feet, but they have similarities.
Right. That's a really good point. Yeah. So, we can record. So, processing information in the brain is, there are many different brain areas involved in this, right? It's not just motor cortex. So when we're thinking about moving to the right, it's not just motor cortex. There's premotor cortex, there's parietal cortex, right? And as we go sort of back, so we have the arm, right? Spinal cord, right? We have motor cortex, and then we can keep going higher and higher in the brain, right? And all of those different areas will reflect or be indicative of the upcoming movement in some way, right? Whereby the motor cortex may be more involved in implementing the details of the movement, whereas parietal cortex may be more involved in sort of a higher level concept of space, right? Of, just maybe rather than exactly how you're going to get there, just the destination or something, right? So I think you're making a really good point that, we can combine information from different sources, and those sources each have different types of information, right? And so, yeah. So now I'm glad you're mentioning this. So we've done work, right, where we've combined these different sources of information, and I think that there's still a lot more that could be done. And I think one of the things that we're going to be able to leverage is now not just recording from one brain area, but now we can record from multiple brain areas at the same time. And this is something that people have not done traditionally, and only in recent years have people been doing more of that. All right, cool. Enjoy the rest of your semester.
Maybe I'll be doing this for my master's or something.
Okay. In regards to neuroscience?
Not necessarily neuroscience. It's the idea of using online learning or kind of cooperation between systems.
Yeah, yeah. I would imagine that machine learning, there's people working on this. The names off the top of my head, but
Okay. All right.
have you checked out the faculty website on machine learning?
A little bit. I'll look into it more closely though.
Okay, sure.
I'm wondering, because animals, the difference between animals and humans have, for example, the